All right, so chapter 7 is going to talk us about the cell membrane and how it's kind of fluid and some of the models that make it up and some of the functions that it performs. And so the first thing I want you guys to do though is to take a look at this TOK question. It says the explanation of the structure of the plasma membrane has changed over the years as new evidence and ways of analysis have come to light. Under what circumstances is it important to learn about theories that were later discredited? So take a few minutes and think about that, write down an explanation, then we'll look it over and talk about it in class, okay? So the main ideas of chapter 7 talk about the structure of the biological membrane makes them fluid and dynamic. In other words, they're not this rigid, unchanging structure. They're constantly kind of moving around and adjusting with their environment. And also membranes control the composition of cells by active passive transport. In other words, what's coming in and out of the cell is governed by the proteins that make up the membrane, how the membrane behaves, and really the things that the cell is able to get rid of, the things that it's able to bring in, those are the things that are controlled by the membrane. Okay, so a good quote here says, "We are made of cells and of stars. The universe outside of us has made the universe inside of us." So in other words, all of the atoms and the different kind of composition of the cells can be found everywhere in the universe. So we're just kind of a little microcosm, I guess you'd say, of the actual living universe. Okay? So here's a little video that I want you guys to take a look at. So have a look at this real quick.
The plasma membrane is made up of several organic molecules. I will concentrate on two of them in this presentation. The first molecule is a lipid. It is called a phospholipid because instead of the usual 3 fatty acid chains, there are only 2. The third bonding position is with a phosphate group. The phospholipid molecule is divided into a head region and a tail region. The head region is the glycerol and phosphate group. The tail region is the fatty acid chains. The head is hydrophilic, attracted to water. The tail is hydrophobic, repelled by water. It is this property which allows for the unique molecular alignment that makes the plasma membrane. If a droplet of phospholipid was put into water, a mycel would form. A mycel is a sphere with the hydrophilic heads on the surface, the tails inside, away from the water. Our bodies are made up of approximately 60% water and trillions of cells. The water can be found inside the cells, outside the cells, and in the bloodstream. The location of water and the phospholipid, hydrophilic, and hydrophobic ends enable the membrane to form a bilayer of lipids. The currently accepted model of the cell membrane was developed by Singer and Nicholson in 1972 at the University of California, San Diego. Pictured here, the plasma membrane looks like a sea of lipid with icebergs floating in it. The membrane is very flexible and has a fluid motion. This design of the plasma membrane is called the fluid mosaic model. Quite often, these proteins have sugar molecules attached to them, forming glycoproteins. These molecules extend from the membrane surface and act as an identification marker. The proteins are used by the immune system to identify a cell as being self, or foreign, called non-self. Each individual has their own biochemical makeup. In other words, each human being has their own set of cellular proteins. But if a cell comes into the body that has a different set of proteins, whether it is a bacteria or an organ transplant, the immune system may identify it as self or as non-self and attack.
Okay, so that kind of gives you an idea of what the cell membrane does, some of its functions other than just allowing things to come in and out. All right, so let's see here. Continue on looking at the cell membrane now. You can see that it forms that bilayer, and this is kind of what it looks like. And it's got 2 layers, of course, which are composed of phospholipids. And the bilayer itself has to really separate 2 aqueous environments like they talked about in the video. So water inside and water outside. And one of the things that we discussed in class was the idea that you have the phosphate part, which is hydrophilic because of the attachment of the choline, the phosphate group, and so forth to it. This is what enables it to interact with water. And then you have the lipid portion, the 2 fatty acid chains kind of facing inward to each other. And that's kind of what's forming that interface between the watery environment of the inside of the cell and then the exterior of the cell, of course. And then one of the other things that we talked about in terms of the structure of these molecules is that you've got this glycerol head, and then to that you've got a choline and a phosphate, which makes it hydrophilic. And then instead of being a triglyceride like we discussed in chapter 5, we actually have a diglyceride where you've got those two fatty acid chains kind of extending inward. So it's just a variation of something that we talked about when we were looking at the triglycerides in chapter 5. Okay, so the plasma membrane itself now, kind of embedded within this, are the protein molecules. And these are things that are involved in selectively transporting certain things back and forth across the membrane. And we refer to the lipid bilayer really as a fluid mosaic, as we discussed in class, because it contains cholesterol molecules, it contains protein molecules, both peripheral types of proteins, which we'll get to in a little while, as well as these integral or transmembrane proteins, which we'll also get to. So it's just a mosaic, as you would see like in an art class or something like that. It's just a bunch of things kind of put together to form this structure. Okay, now the evolution of the cell membrane, this was one of the things that we spent some time on in class, just kind of discussing the whole idea of the during the 20s you had Gorder and Grendel that had kind of come up with the idea of the phospholipid bilayer, and then in the 30s you had Davison and Dinelli kind of extending some of this work, and that work really lasting in excess of 30 years until Singer and Nicholson kind of came along and came up with our current model, which is the fluid mosaic model of the actual lipid bilayer. Okay. So Gorder and Grendel, we talked about them using red blood cells to basically show that, and what I used when we explained this is if you look at my hand and you see this, you'd say that it's got a certain amount of membrane to it, but when you actually break it apart, you see that the membrane is double, so it's got an inside and an outside to it, so the lipid content is really double of what you would expect if you were just looking at it, and that kind of suggested it was a bilayer. And they also concluded that what it was doing was it was keeping the inside different from the outside. And of course, they were looking at red blood cells, which we talked about. And they figured that the phosphate portion of this bilayer must face outward because the ability of it to interact with the water, and of course the lipid, the fatty acid chains kind of face inward within this bilayer because of course they're hydrophobic. Okay. So here's what their model kind of looked like, and if you look at that, this is get my pen up here. So this portion, of course, there we go. Well, this thing is not cooperating right now, sorry. So this portion right here is the phosphate head, of course, and then these portions right here, these are the fatty acid tails. So this would be, in here, this would be the hydrophobic region of the lipid bilayer, and then the yellow region out here and over here, these would all be the hydrophilic portions, okay? So continuing on, when we're looking at this type of thing, Davison and Dinelli kind of extended some of the work of what they were doing, and once they determined that it was a bilayer, the big question became, where do you place these proteins, and why do you place them there? And they came up with what was known as the sandwich model, which we talked about, because what you've got is this layer of lipids in between two layers of proteins, because they found that the proteins actually interact with the water much better than the kind of phospholipid heads, so to speak. And this is kind of what came up with their hypothesis. So if you look at this model right here, we can see these purple proteins, okay, right here, all throughout the top and the bottom of this layer. And then you had the phospholipids, of course, which are yellow right through here. And then you had, in this region right here, you had these fatty acid chains. And that was kind of the model that held up for quite a while because of the experimental evidence that kind of supported this type of thing. It was supported by the electron micrographs, which we said the electron microscope was recently invented and people were learning how to use it. And the thing was, though, is as time went on, people got more adept at using these technologies, and the technologies themselves improved, and they started to find that this model didn't really fit for reasons we discussed in class, that a good model should cover most, if not all, of the things that you're talking about. And this sandwich model really only kind of supported a few different types of things. So more research was showing that perhaps the model needed to change a little bit. There were different types of membranes that had different compositions, for instance, and their solubilities when mixed with water were a little bit different. There were different membranes that had different thicknesses, and none of these things really matched kind of on the whole, which is what a good model should do. And one of the examples was the mitochondrial membrane was much thicker, or excuse me, much thinner than the actual plasma membrane, had a different appearance, a different composition. So if we're looking at different types of membranes, our model should try to take into account as many of these as possible. Okay, so this kind of information, this experimental information and so forth, led to kind of the downfall of the sandwich model. And there was more kind of evidence revealed through the electron micrographs that were being developed from this as that technique improved. And what they found was, like we said, with the phospholipids have an amphipathic appearance. In other words, they have the ability to bind with water as well as kind of fear the water. So they're hydrophobic and hydrophilic portions. They found that the proteins had this phenomenon as well. Some proteins had hydrophilic regions, whereas the same protein also had hydrophobic regions. And we referred to this, of course, as being amphipathic. Okay, so then in the 60s, Singer and Nicholson started to kind of develop their fluid mosaic model, and they presented it in 1972. And what they hypothesized is that you had a series of proteins that were kind of embedded within this membrane, within this bilayer, instead of being kind of on the outsides of it. And the hydrophobic regions were embedded within the membrane, okay, interacting with the other hydrophobic portions of the actual lipid bilayer. And the hydrophilic regions were found inside and outside of the cell. So if we were to look at this, we can see, like, for instance, in this region right here, this would be hydrophobic, and then these outer regions right here would be hydrophilic. So that's the amphipathic nature of these types of molecules. And the model was good because it supported the types of things that were being seen now as the techniques of using the electron micrographs or the electron microscope, were improving, such as the freeze fracture. Even though that kind of information was used to support the Davison-Donnelly model, as they became better at it, they started to notice certain things with freeze fracturing that kind of fit this fluid mosaic model a little bit better. And we talked about striking a small portion of tissue with a very sharp kind of diamond type of a blade and getting it to split apart or almost peel apart, as you're seeing here. And you can see these are the proteins that are kind of embedded within this membrane. And then instead of being on the top and the bottom, and then you have this kind of lipid bilayer here, and then this lipid bilayer here that's being separated apart. And as you look at the electron microscope images down here, you can see that they're kind of fitting the freeze fracturing type of pictures that we're seeing with the electron microscope. So looking at it a little bit further, you can see that the nuclear membrane, which is a form of a membrane, this has pores in it, like you would see if we were looking at this type of a structure. Some of these proteins themselves are forming these pores that you're seeing. So when you look at the transmission electron micrograph, kind of looking through it, like we see up here on the top, what you've got is little pores that are forming right here that are allowing things in and out of the cell. And then there's regions here that don't contain any pores. And then when you look at it a little bit differently through a freeze fracture, here are all these little pores that we're noticing up above, okay, right in through here, for instance. And these are the types of things that we're starting to be able to see, because as you can see, this top figure, 120,000 times magnified, the bottom figure over 250,000 times magnified. So as these techniques started to improve, they're starting to support the Singer-Nicholson model, the fluid mosaic model, rather than the earlier models. And what this fluid mosaic model kind of proposed was the idea that the fluidity of the membrane is variable. It's always kind of changing. The lipids are moving around very rapidly. The proteins, some of them move quickly with the lipids, and others seem to be kind of anchored in place to the cytoskeleton that's there. So the model that they were developing was being supported by the experimental findings. Okay, so what I want to do now is show you another video here, just kind of look at this and summarize a little bit of the stuff that we've talked about.
Every cell in your body is separated from those around it by its outermost layer, its membrane. A cell membrane must be both sturdy and flexible. Imagine a membrane made of metal, great at keeping the cell's guts inside, but horrible at letting materials flow in and out. But a membrane made of fishnet stocking would go too far in the opposite direction, leaky but easily torn. So the ideal membrane falls somewhere in the middle. Over the past few centuries, we've learned a lot about the way membranes work. The tale starts in the late 1800s. when, according to legend, a German woman named Agnes Pockels was doing dishes. Her observation that not all detergents dissolve grease in the same way piqued her curiosity, so she made careful measurements of the size of soapy films that formed on the surface of a metal tray filled with water. Later, in the 1920s, GE scientists Irving Langmuir and Kathleen Blodgett re-examined the problem with a more elaborate contraption and found that those tiny slicks were in fact a single layer of oil molecules. Each oil molecule has one side that loves water and floats on the surface, and one side that loathes water and protrudes into the air. So what does it have to do with cell membranes? Well, at the turn of the 20th century, chemists Charles Overton and Hans Meyer demonstrated that the cell membrane is composed of substances that, like oil, have a water-loving part and a water-loathing part. We now call these substances lipids. In 1925, two scientists, Evert Gorder and Francois Grendel, pushed our understanding further. They designed an experiment meant to test whether cell membranes are made of only one layer of lipids, a monolayer, or two layers stacked on top of one another, called a bilayer. Gorder and Grendel drew blood from a dog, a sheep, a rabbit, a goat, a guinea pig, and human volunteers. From each of these samples, they extracted all the lipids from all the red blood cells and placed a few drops of this extract on a tray of water. True to form, the lipids, like oil, spread out into a monolayer, whose size Gorder and Grendel could measure. If they compared the surface area of that monolayer to the surface area of the intact red blood cells, they'd be able to tell whether the red blood cell membrane is one or two layers thick. To understand the design of their experiment, imagine looking down at a sandwich. If you measure the surface area of what you see, you'll get the dimensions of a single slice of bread, even though there are two slices, one stacked perfectly atop the other. But if you open the sandwich and place the two slices side by side, you get twice the surface area. The Gorder and Grendel experiment is basically the same idea. The open sandwich is the monolayer formed by the extracted cellular lipids spreading out into a sheet. The closed sandwich is the intact red blood cell membrane. Lo and behold, they observed a 2-to-1 ratio, proving beyond a shadow of a doubt that a cell membrane is a bilayer, which when unstacked yields a monolayer twice its size. So almost 30 years before the double helix structure of DNA was elucidated, a single experiment involving fancy versions of household materials enabled deep insight into the basic architecture of the cell.
All right, so that kind of explains some of the history behind the cell membrane and talked about a few of the things that we've discussed. And you don't need to know all of those people. Just worry about the ones that were kind of in the presentation here that we talked about. And then, we started with Gorder and Grendel, then we talked about Davison and Donnelly, and then Singer and Nicholson. So those are the ones that you really want to concentrate on, okay? So Singer and Nicholson kind of came up with this fluid mosaic model, and you can look kind of here, and you can see these peripheral proteins, the ones that are on the outside. You can see these transmembrane or integral types of proteins that kind of span through the entire membrane, and then you can see some of the other structures that are on there, the glycolipids, glycoproteins, all of those sorts of things. And what these proteins basically do is I guess you could say, is they fall into two categories. They're either integral, or we refer to those as transmembrane sometimes, where they're spanning completely across the plasma membrane, the bilayer. And then the other types of proteins are peripheral proteins. And these are either found on the surface of the outside of the protein or the surface on the inside of the protein. And they do not span all the way through. So there's two classes, those that span all the way through and those that are either found on the inside or the outside. Okay? And there's 6 main functions to the membrane proteins, and we're going to talk about these in the next part. All right? So if you have any questions, like always, make sure you write them down, and we'll discuss them in class. We'll talk to you soon.